Let's now start with the nerve physiology, for which you should know what a nerve is. Well, a nerve is a bundle of peripheral axons. Now, let me give you some basic concepts. Nervous system is composed of neuroglia and neurons. The neuron is the name which is given to the nerve cell, including all its processes. And it is the basic structural and functional unit of the nervous system. Human nervous system, it has about 1 trillion of these cells. And the neurons, they are highly differentiated and specialized excitable cells, and their specific activity is the reception of stimuli, the generation of nerve impulses, and their transmission as well to the other nerve cells and to the effectors, which may be the muscle fibers or glands. In general, each neuron, it possesses a nerve cell body which is called soma, and two types of processes, the dendrite and the axon. And these processes, they are also called neurites. Mm, while as a rule there is always only one axon, but the number of dendrites may be one or more. The axon, it has been defined from a physiological point of view as the process that carries the nerve impulse from the nerve cell body. And the dendrite is defined as the process that brings the nerve impulse to the cell body. Now, these axons, they are also commonly called nerve fibers as well as axis cylinders. The bodies of the nerve cells, they lie either in the nuclei within the gray matter of the central nervous system or in the ganglia present outside the central nervous system. For example, in ganglia of, say, posterior spinal roots, cranial ganglia, and the ganglia of the autonomic nervous system. You see, a ganglion is a group of nerve cell bodies which are present somewhere outside the CNS. But keep in mind that basal nuclei, they are present within the cerebrum and they are still called basal ganglia by some authors. Well, nerve cells and the fibers, they are completely enclosed by special supportive and nutritive cells which are called stellate or the glial cells. Neuroglia of the peripheral nervous system, it completely surrounds the axons and the cell bodies of neurons. And it is like a glue that holds the nervous tissue together. And in addition, it actively participates in the activities of nervous tissue. Neuroglia, they are different cells and they are divided broadly into microglia and macroglia. Microglia arises from macrophages, and they function to remove the cell debris and the damaged cells as a result of injury, some disease, or infection. If we look at the classification, macroglia are large cells and they are of three types. oligodendrocytes, Schwann cells, and astrocytes. Now, the oligodendrocytes, they take part in the formation of myelin sheath inside the nervous system. While the Schwann cells, they form myelin sheath in the peripheral nervous system. What do the astrocytes do? They form tight junctions and hence they have a role in the formation of blood-brain barrier, and they also take part in the formation of myelin sheath. So that was a brief introduction to the neuroglia, and now we move on to the neurons. Uh, you must know that neurons, they are electrically excitable, and they communicate with one another using the electrical signals. A nerve fiber, it's a general term for any neuronal process or extension that emerges from the cell body of a neuron. The nerve cells, they can be classified in three ways: histologically, physiologically, and according to their size. But of course, I'm going to focus on the physiologic classification only. So, physiologically, the neurons could be motor, sensory, or internential. Now, the motor neurons, they are also called the effector neurons, and these neurons, they send impulses to the effectors, which may be muscles or glands. The sensory neurons, they are the receptors, and they receive afferent yani incoming fibers, which are responsible for various sensations, for example, touch, pressure, pain, temperature, light, taste, smell, sound, etc. Now, what about the internential neurons? These are basically the interneurons, and these neurons, they are also called the connecting neurons or the contact neurons, and they lie within the central nervous system, and they connect the receptors and the effector neurons. And according to the character of the effect which is produced by them, these are called either excitatory or inhibitory neurons. Well, moving on to the structure of neurons, this structure can be divided into easily into three parts: the soma, uh, dendrites, and axons. So, soma is the nerve cell body, and it comprises the nucleus and the surrounding cytoplasm. The nerve cell body, it may be of several shapes. For example, it could be fusiform, stellate, oval, rounded, or even pyramidal. The size greatly varies from about 5 microns, and when I say micron, it means micrometer, from 5 to even 130 microns in diameter in case of the granule cells of the cerebellum and the alpha motor neurons of the spinal cord, respectively. So, the nucleus, that is typically large, and it possesses usually one nucleolus. Sometimes there may be more than one nucleoli. Nerve cells, they possess a single nucleus, except those of the sympathetic and the sensory ganglia, which are binucleated. The cytoplasm, it has mitochondria, ribosomes, endoplasmic reticulum, lysosomes, and Golgi apparatus. These components, they are found in other body cells also. The nerve cell cytoplasm, however, it contains some characteristic components which are called nasal bodies, or the nasal granules, or you can call them the chromatic bodies. And then they also have the neurofibrils. The nasal bodies, they are composed of RNA and the polysomes, and they are stained with the basic dyes. And they are also called the tigroid substances, the tiger like substance, due to their striped appearance, and they represent ribosomes. These occur in the dendrites and in the perikaryon, mean around the nucleus area only, and not in the axon. Now, in addition, the part of the perikaryon from which the axon arises, and this is called axon hillock, that is also devoid of nasal granules. Okay, if we talk about the dendrites, dendrites are short, tree like, highly branched, tapering processes of the nerve cells. In general, the nerve impulses, they travel from the dendrites towards the cell body. The dendrites, they are often highly branched, thus enabling the cell body to receive impulses from a large number of other neurons. These impulses, they may be excitatory or inhibitory. In certain places like cerebral cortex and cerebellar cortex, the dendrites, they show small knob-like projections which are called dendritic spines. The dendrites, they have all the components present in the cytoplasm of the nerve cell body. Now, axon is the name which is given to the longest process of the neuron, and this is also called the axis cylinder or the nerve fiber. It is usual to state that an axon always conducts the impulses away from the cell body. This is true only for the motor axons. The axons of the spinal posterior root ganglion cells, they are an exception, because in this case the longest neurite, it carries the nerve impulse towards the nerve cell body. However, barring this exception, the term axon that is used for the longest neurite that conducts the impulse away from the nerve cell body. In short, we can say that a neuron, it consists of a cell body and two types of processes, the dendrites and the axons. The cell body, that is variable in size and shape, and it has a nucleus, and the cytoplasm, it contains all the cell organelles except the centrioles. So, should I recall that now what are these nasal granules? You see, the nasal granules, they are the membranous organelles which are found only in the neurons, not in any other cells of body, and they are composed of the RNAs and the proteins. And where can we locate these granules? They are present in the cell body and the dendrites, but not in the axon. Uh, they're stained by the basic dyes and named after their discoverer. Uh, the interesting bit is that they are also called the tigroid substances, as I already told you, since these bodies, they are responsible for the tigroid or the spotted appearance of the soma after suitable staining. Number and size of the nasal bodies that depend upon the functional state of neuron. The nasal granules, they flow into the dendrites from soma, and the axons, they are distinguished from the dendrites under microscope by the absence of nasal granules. While I am at it, I mustn't forget to mention chromatolysis. You see, fatigue and injury, it results into breakdown of nasal bodies, and this is called chromatolysis. Although the granules, they reappear after recovery or regeneration of the nerve fiber. The nasal granules, they are broken down and then they undergo chromatolysis when the axon of a nerve cell is cut. The same change, it is also seen when the nerve cell is injured or is fatigued or subjected to certain poisons and ischemia. The nasal substance is responsible for synthesizing proteins which flow along the dendrites and the axon to replace the proteins which have broken down due to cell activity. And the neurofibrils, they run parallel to each other, and they are seen to be continuous in the dendrites, cell body, and the whole length of the axon. Well, moving on to the processes of the neurons, the dendrites, they are short and branching, and they may be one or more than one. But the axon, that is always one, and it is devoid of the nasal granules. Now it arises from an elevated and tapered portion of the cell body, which is called axon hillock. The length of axon, that is also variable, and the length of the longest axon, it could be up to 1 meter. The axon, it emerges from the cell body at the region which is termed as axon hillock. Now, the first 100 microns or so of the axon, they have no myelin sheath even in the myelinated nerve fibers, and along with the axon hillock, it constitutes what is called the initial segment. And the characteristic property of the initial segment is its very high excitability. Axon has a long central core of cytoplasm, which is called exoplasm, and it is covered by exolemma. So the exoplasm, along with the exolemma, that forms the axis cylinder of the nerve fiber, and this axis cylinder, that is covered by neurolemma. At the end, the axon, it gives rise to a number of the terminal branches which are called the synaptic knobs or the nerve terminals or even ant feet. Well, the axons, they do not branch as profusely as the dendrites, but many axons along their course, they show side branchings which are called the collaterals. The axons and their collaterals, they may remain within the central nervous system or they pass out into the peripheral nerves. Those which end within the central nervous system, they do so by breaking up into a number of terminal filaments or the telodendria. And at the end of the telodendrion is a small swelling which is called the button terminox or the terminal button, or you can call it a synaptic knob. Now, these knobs, they contain the granules or the vesicles in which the synaptic neurotransmitter substance that is produced by the nerve cell that is stored here. So, in the synaptic knobs, mitochondria and a number of the vesicles they are present, and the vesicles they contain the neurotransmitter. In the body, a nerve fiber it carries impulses in one direction only. If a fiber it carries impulses from the CNS to the periphery, it is called efferent. While a nerve fiber which is carrying the impulses from the periphery toward the CNS, that is called afferent nerve fiber. However, if a nerve fiber is isolated and it is stimulated anywhere along its length, then the nerve impulse it travels in both directions. Let's now talk about the myelin sheath. A multi-layered lipid and protein covering surrounding the axons is called myelin sheath. And nerve fibers, they may be myelinated or unmyelinated depending upon the presence or the absence of this myelin sheath. The myelin sheath, it consists of compressed layers of cell membrane of Schwann cells concentrically wrapped around the nerve fiber. The composition of the myelin sheath, it is mainly lipids and proteins, just as in the cell membranes. Now, the uh, nerve fibers which lie outside and within the CNS, they could be myelinated or unmyelinated. And there is a fundamental difference between the nerve fibers lying outside and inside of the central nervous system, that is, in respect to the myelin sheath which is surrounding them. The nerve fibers which lie outside the central nervous system, they are enveloped by specialized cells which are called Schwann cells, and they provide uh, both the structural and the nutritive support. So, depending upon the histological characteristics, the nerve fibers they may either be myelinated or unmyelinated. In case of myelinated nerve fibers, the Schwann cell that is invaginated by a single nerve fiber so that the fiber is embedded in a groove in the Schwann cell cytoplasm. The plasma membrane of the Schwann cell it fuses along the opening of the groove, thus effectively sealing the nerve fiber within an extracellular compartment within the Schwann cell. Now, the unmyelinated nerve fibers outside the central nervous system, they are the nerve fibers of small diameter which are present outside the central nervous system. And for example, the fibers which are conducting pain impulses and those belonging to autonomic nervous system. Um, and the example is postganglionic sympathetic nerve fibers. So these represent the unmyelinated nerve fibers outside the central nervous system. While the nerve fibers which lie within the central nervous system, they are also of two types, the myelinated and unmyelinated. The myelinated nerve fibers, although these fibers they show a myelin sheath, uh, just as the myelinated fibers outside the central nervous system, but the myelin sheath here, that is produced not by the Schwann cells but by specialized glial cells which are called the oligodendrocytes. And these cells, they are present along and between the axons, and thus they are the counterparts of Schwann cells present in the peripheral nerve fibers. Now, within the central nervous system, the unmyelinated nerve fibers, unlike the myelinated ones, they are not supported by the specific cells, means the oligodendrocytes, but they are indirectly supported by the mass of the surrounding tissue. Let's now talk about the gaps in the myelin sheath, which are named the nodes of Renvier. Actually, the myelin sheath, it is not continuous around the nerve fibers. It is absent at the regular intervals, and these areas, they are called nodes of Renvier. In case of myelinated nerve fibers, the myelin sheath, it is not a continuous sheath, but it is deficient at the regular intervals. And at these places, the neurilemma, it dips inwards and give rise to the formation of the constrictions, which are called the nodes of Renvier. These nodes, they are close to each other, and the maximum internodal distance, that is only 1 millimeter. And the branching of the axon in case of the myelinated fibers, that always takes place at the nodes. In case of myelinated nerve fibers which are lying outside the central nervous system, a single Schwann cell, it gives rise to the myelin sheath of each internodal segment. Thus, the cells of the Schwann, they are not syncytial. The cells of the Schwann, they are important in the repair of the nerve fibers after damage. And the nerve fibers in the brain, they are without these cells, and therefore their regeneration is not possible once they are destroyed. The myelin sheath, along with nodes of Renvier, that is essential for the fast conduction of saltatory type of the nerve impulse. If the myelin sheath that is not developed, or it is destroyed, the axon will not conduct the impulses in a normal manner. For example, the destruction of the myelin by the antibodies against it, that results in a disease which is called multiple sclerosis. The outermost layer of the nucleated cytoplasmic myelin sheath of Schwann cells, that is called neurilemma, or it is also called neurolemma or the sheath of Schwann. A neurolemma is found only around the axons in the peripheral nervous system. At the node of Renvier, only neurilemma is present. So when an axon is injured, the neurilemma, it adds for the help in the regeneration. Keep in mind that since neurilemma, it is absent in the central nervous system, so if the nerve fiber is damaged within the central nervous system, it cannot be regenerated. So, if there is a question that which functions are performed by the myelin sheath, then keep in mind that the myelin sheath it acts as insulator, which means that it is not permeable to ions, and then myelin sheath it increases the velocity of the conduction of nerve impulses. The conduction of action potential in the myelinated nerve fibers in the jumping manner from one node of Renvier to another, that is called saltatory conduction. And this special type of conduction, it has certain advantages. Just say velocity of the transmission that is increased, and it also conserves the energy as only the nodes they depolarize. Now there are three connective tissue coverings of the nerves: the endoneurium, perineurium, and epineurium. Endoneurium is present around a single nerve fiber, while perineurium surrounds the nerve bundle or the fascicle, and epineurium that is around the whole nerve trunk. Uh, Here, if you can see, the endoneurium that is a thin layer of finely reticular connective tissue fibers that lies just outside the neurilemma and surrounds the individual peripheral nerve fibers, separating them from each other. This layer persists indefinitely even when its axon is degenerated. And in that case, it forms a tube which is called the endoneurial tube, through which the regenerating axon it can pass down in the regeneration stage. In the case of nerve trunk, which contains hundreds of nerve fibers, bundles or the fascicles of several nerve fibers, they are seen surrounded by a layer of the connective tissue which is called perineurium, and the nerve trunk itself that is surrounded by a loose layer of elastic tissue and the connective tissue which is called epineurium. Genesis, that means the formation of myelin sheath, and for that, Schwann cells they form the myelin sheath around the peripheral nerves. There are many layers of myelin sheath which get compressed. Uh, you see, no matter how cute they look, but did you ever wonder that why babies are so clumsy? Because, my dear student, when a baby is born, his or her nerves they are not completely myelinated, and this completion it may take about 2 years. Now, the examples of myelinated nerve fibers they are the autonomic preganglionic fibers and somatic nerve fibers having a diameter greater than 1 micron. These examples they could be your MCQ option. The examples of unmyelinated nerve fibers they are autonomic postganglionic fibers and the somatic nerve fibers which are having the diameter less than 1 micron. Well, the axonal transport is the transport of vesicles containing neurotransmitters from the cell body to the nerve terminals. And that takes place, this takes place by the exoplasmic or the axonal transport. This axonal transport, this is of two types. It could be anterograde and then it could be retrograde. The anterograde or the orthograde transport that occurs from the cell body to the nerve terminal, while the retrograde transport that occurs from the nerve terminal to the cell body. You should know that the axon, it consists of a central core of jelly-like semi-fluid substance which is called exoplasm, and this is surrounded by a membrane called the exolemma. Now, the exoplasm that is derived from the cell body, which continuously synthesizes new material needed for the growth and regeneration of the exoplasm that flows into the axons centrifugally. And this flow occurs at a rate of about 1.5 millimeter per day. Enzymes which are needed for the synthesis of neurotransmitters, they move centrifugally at a much higher rate. And certain substances, they move into the reverse direction, which means from the axon terminals towards the cell body. And in that case, that is called the retrograde flow. And in the retrograde flow, uh, that transport the proteins, nerve growth factor, tetanus toxin, and certain neurotropic viruses such as rabies and poliomyelitis. Even some used up synaptic vesicles, they reach the cell body and they undergo recycling. The exoplasm, it contains the neurofibrils continuous with those in the cell body and the dendrites. These neurofibrils, they run parallel to the axis of the fiber and they are responsible for the longitudinal striations. Of these two subcomponents of the neurofibrils, means the neurotubules and the neurofilaments, actually it is the neurofilaments whose number that determines the thickness of an axon. Mitochondria and the endoplasmic reticulum, they are also present in the exoplasm, but as I already told you, it does not contain the nasal granules, the Golgi apparatus, and the ribosomes, so it cannot synthesize the proteins.